Nihira,
Yes.
may I request you to just get Professor Amir Bagchi, because otherwise, if he's not online, he'll call me right through the.
Sure.
Yeah, Nihira, you carry on. I'll take care of it.
Yeah.
He'll get quite agitated if he's not, and if he's not well connected.
He responded this morning saying that to my link, I had resent it.
He
And he
might
responded it.
I don't want to disappoint him. Yeah. Okay. Yeah. Ah, there he is. Good evening, Professor Bagchi.
Hello, Amyoda. Hello, Amyoda.
Hello. So now you can't blame us for not connecting you now. From you?
So the webinar has opened up to attendees. We'll be beginning in 2 minutes. Okay, so we can begin now. Okay, that's okay. So hello everyone. Welcome to the second lecture in the lecture series organized by the Foundation for Agrarian Studies in collaboration with Rosa Luxemburg Stiftung South Asia. The series is on science and technology in agriculture in India. And it was a 4-week series that we had planned, which began with an inaugural public lecture delivered by Professor Ramkumar and chaired by Professor Madhura Swaminathan. Our first lecture was on seed science by Dr. Rajendra Prasad, who is the vice chairperson at the University of Agricultural Sciences, Bangalore. I'm happy to present to you our second lecture of this series. It will be delivered by Dr. K C Bansal, who has served as the coordinator of National Project on Transgenics in Crops, ICAR, and also as the director of the National Bureau of Plant Genetic Resources, ICAR, in New Delhi. Dr. Bansal will be speaking on transgenic and CRISPR technology and its use in agriculture in India. The lecture today will be chaired by Professor V.K. Ramachandran, who is the managing trustee of the Foundation for Agrarian Studies. He is also the editor of the biannual journal Review of Agrarian Studies and has contributed to and edited various publications such as Women in Work in Rural India and Dalit Households and Village Economies. I now request Professor V.K. Ramachandran to carry on the proceedings of today's lecture. Thank you.
I'm sorry, my video is off. I'm sorry. Thank you, Nihira, Professor Bansal, and colleagues. Welcome to this, the second lecture, the third lecture in the series, in our series on science and agriculture, science, technology, and agriculture. This is, as Sandeepan mentioned before this meeting started, we have an annual workshop for students and young people, at which we have a theme subject. Last year, we did not hold it because we were not yet familiar with, we were not yet tuned into the Zoom technology, and which has become, it seems strange that now it's become second nature to all of us. So this year we're holding it on the same topic as we intended last year, on science and technology. The overall, we at the Foundation for Agrarian Studies, although we are primarily social scientists, though some with an agricultural background, we are keen students of the role of science and technology in agricultural development. We recognize that agriculture is central to the survival of humankind, for the provision of food for all people, and for the fulfillment in different degrees of the needs of people with regard to food, fiber, and also, in addition to food, fiber, fuel, and construction materials. And although there is it would have seemed obvious about, say, 20 years ago, 25 years ago, to say that agriculture needs agricultural science and technology need constant advance, both to meet the new challenges of climate change and ecological pressures, and to provide nutrition for the malnourished. In other words, agriculture not only has to meet climate change and ecological pressures, but also to meet the needs of an increasing population for nutrition itself, for food itself. Now, it's worth progress in agricultural science and technology is necessary if agriculture is successfully to meet these needs by, say, 2050, when the population of the world is estimated to be around 10 billion. And please remember that it's worth recalling that in 1968, in the early years of the Green Revolution, the population of the world is about 3.5 billion. And so there's going to be an enormously increased demand. And this is not merely, it's not a banal repetition of the need for science in agriculture, because there is a fairly strong element in social science today, which in fact says that the challenge of the 21st century is organization and distribution and empowerment, not science, technology, and actual production enhancement. So this is something which the Foundation for Agrarian Studies, it's an argument that we want to face head on. That to that extent, we are openly challenging the argument that you don't need enhanced production. You don't need enhanced science and technology. What you need is better social and perhaps human organization. That is inadequate, that is sufficient. That social science or social science solutions or policy solutions are not merely necessary condition, but that they are sufficient conditions. Now that is a position that we challenge. In recent years, therefore, the application of science and technology to agriculture and even research and field trials has been under sustained attack from very often well-meaning groups, individuals, etc., who profess commitment to the protection of the environment, to sustainable agriculture, and to the enhancement of the livelihoods and of working people and rural poor. Now, these goals, I must assert, and all our work in FAS underlines that these goals are universally shared among conscientious people. But it is the means towards achieving these goals of sustainable agriculture and the enhancement of incomes, livelihoods, and food supplies. It's the means to that objective that generate division. In recent times, the targets of persistent and widespread attacks on science and technology are, inter alia, all seed material produced off-farm, all synthetic chemicals used in agriculture, whether for soil nutrition or plant protection. A host of modern farming practices, including mechanization, and more recently, farming practices that involve any use of fossil fuel, any medium or large-scale irrigation schemes, and different methods of the exploitation of groundwater. At different points, all of these have been under attack. Now, our concern here is not with sincere worries about the careful deployment of new technologies or the destruction of agrarian heritage. Our concern is with those whose opposition to science and technology is hostile to the very method of science itself. Such criticism is very often based on ephemeral measures of public opinion, or anecdotes, or hearsay, or surveys which are faulty or characterized by ignorant methods. And it is to engage with arguments against the application of modern science and technology, in which, as I said, the production of seed material is now occupying very often a central place. That we have organized this series. So there could be no better person. Today, Dr. Bansal is going to speak to us on transgenics and CRISPR technology in agriculture, and we couldn't have a better person to speak to us on this. In fact, Dr. Bansal presents a problem to any chair that if you start reading out his achievements, we'll cut seriously into the time. So time available to us. So let me say that Dr. Bansal comes from the Indian Agricultural Research System. He's a gold medalist and doctorate from the IARI. He did his postdoctoral work at Harvard. He was the coordinator of the national project on transgenics in crops. He was a director and achieved great eminence as the director of the National Bureau of Plant Genetic Resources in New Delhi. He was the first Norman Borlaug Chair of the ICR for crop improvement. He is currently the Secretary of the National Academy of Agricultural Sciences and is a member of the Board of Directors of the Global Plant Council and on the editorial board of Plant Biotechnology. His basic interests include genome engineering and functional genomics for enhancing abiotic stress tolerance in crop plants. He has published very widely. He's won many awards in the field. And he's a fellow of both the National Academy of Agricultural Sciences and the National Academy of Sciences, of the NAS as well, of NAS and NAS in India. Thank you very much for having joined us, Professor Bansal. It's a great honor for us. And we really look forward to what you have to say. I personally have been, for the last few weeks, we have been following the new flurry of activity, particularly in CRISPR technology. Japan now has commercially released, Apple has been released in different parts of the world. CRISPR modified apples that resist browning. And Japan now, as of last week, has released three. There are three commercially available products of CRISPR technology. Two are aquaculture products, sea bream and pufferfish, and the CRISPR tomato has been available since last year. So India has more or less missed the transgenic bus, unfortunately, for various reasons. I hope we are able to regain strength there and to at least gain some strengths in the future in the field of CRISPR technology. But this is the subject of your lecture. Thank you very much. Professor Bansal.
Thank you. Thank you, Sir Ramachandran. Good afternoon to all of you. And particularly, I'm thankful to Dr. Ramachandran, Dr. Sandeepan, Madam Madhura, Swaminathan, and others, for giving me this opportunity to talk to you on this particular topic, which is assigned to me. And I'm very happy that the inaugural lecture was delivered by Professor Ramkumar in this series of Science and Technology in Agriculture in India. And today I'll be talking about the application of transgenic and CRISPR technology to continue with the role of science technology, in agriculture, how important it is. And, I'll be giving you briefly kind of a, historical perspective and the methods which have been used in past for crop improvement particularly. And then mostly my talk will focus on these two technologies, as transgenic and CRISPR technologies. And by end of my presentation, I'll try to convince actually that how important, the science and technology is today in the world, not only in India, for global agriculture and food security and nutrition security, and particularly in the face of climate change, that how we can try to, in fact, not harm the environment, but safeguard the environment, while still addressing the issues of sustainability or meeting the challenges posed by the climate change and malnutrition, particularly in India. So with this, thanks again to the organizers, particularly to the Foundation for Agrarian Studies for this particular opportunity. And I congratulate them to really speaking, initiate this kind of a lecture series, for those young scholars. I'm sure they will try to learn and in process, in the question answer session, probably we'll learn from each other more. So friends, in the first slide, I try to just show you the milestones, which we have crossed, in the last century and continue doing so even in the present century with regard to the methods of crop improvement. See, 10,000 years ago, when the agriculture started, actually speaking, the farmers, they started the process, we call it as crop domestication, when the farmers will try to select the best species out of those crops which were otherwise grown in the wild and they were not high yielding. So whatever the best forms of those crops, they started selecting, from the plants grown in the wild. For example, there's a case of domestication of wheat. Followed by in 1900 with Mendel's laws being rediscovered, the process was further streamlined, and it was like selective breeding or conventional plant breeding, where the cross breeding of those two parents will take place, which have got some good features already in them. This is all actually speaking in a way, artificial selection, we call it, or selective breeding, by human intervention. And then this was followed by in 20s, 1920s, about 100 years ago, of hybrid breeding, where still you cross, of course, two different types of, varieties, but they are supposed to be genetically diverse, distinctly related, so that you can develop better performing hybrids. Here, the example is more vigorous hybrid maize, which was done in case of, in USA, as it's been shown here. I don't know if you can see it. This is the wild species, of maize, which I was referring to here, crop domestication. And the farmers will try to select the best out of them. And over the years, they will every generation to select the best and come to a particular stage. The idea was of selecting either the larger crop size or the more grains and seed number in a particular crop. And then this is something which is shown here is a hybrid maize. You can imagine how far, from this wild ancestor, which is hardly you can just see, on the same scale, to this particular level and increasing production. And, after hybrid breeding in 1940s, the scientists discovered the process of mutagenesis. I will just try to explain briefly in simple terms. Mutagenesis actually is nothing but, so, we're all governed by the DNA, the human beings or all other organisms, including plants we carry. Our features are all governed by DNA. As I go further, you will learn more about it. So idea was to create more of genetic variation, and therefore the mutagenesis came into being, and I'll explain to you more, give you some examples from the Indian work as well. And mutagenesis, by I mean, is developing new genetic diversity or variation by exposing the already, let's say, high yielding crop plants to chemical agents or radiation. If it is a plant is high yielding, you want to add resistance to a particular disease, then you try through this method, which is random mutagenesis, actually speaking, and compared to what we are talking about targeted mutagenesis today. I'll explain it to you more as I go further. So what is then plant domestication? Plant domestication is the process whereby wild plants have been evolved into crop plants through artificial selection with human intervention. And this usually involves an early hybridization event followed by selective breeding. In nature also, these crosses are naturally taking place, but then farmers will be trying to select the best, and that is selective breeding. So selective breeding is when human intervention will take place and they will choose which animal or plant species to be bred together, not only plants, even animals as well. So they choose the plants or the animals from the existing stock with the features you want. See, natural selection is happening where human beings have no control. But if we want to breed the best, then you choose what is that you want, from the high yield point of view, or better taste point of view, or better flavor point of view. So therefore, you choose the plants, or for that matter, animals, from the existing stock the scientists will have, or the farmers will have themselves, and with the features you want. Then you breed these together, with each other, cross both together. And then out of the offspring you get, choose the best, or the better ones you choose. And then continue this process, 1, 2, 3. Continue this process over several generations, and eventually all the offsprings have the features that you're looking for. So it takes about 10 to 20 years, even more, that you continue this process, and you keep on continuing selecting the best out of what you have. But then while you're doing it, you're doing it only on the basis of particular traits. You have no idea what kind of genetic changes are taking place in the plants. They are taking place, but we have no idea so far. Okay, when we're talking of this level of selective breeding here. Okay, and there are certain problems as well with selective breeding, that it reduces the number of genetic variation in a population, a number of alleles. I'll describe it to you more, or genes it reduces. As shown here, if this is a wild species, and in terms of domestication, we have selected, let's say, the best. The farmers have selected what is the best looking out of all these, wild species. And this we call as early domesticates. And what is the problem here is you can see these different color squares and boxes are representing different genes or alleles, as I mentioned here, different genes. So they have, for example, different colors like red, green, yellow, etc. But here you see that yellow is disappeared, because yellow probably did not contribute to the bigger size of the cob or more grains of the cob, etc. So because the farmers wanted to harvest more, for their food, and meeting their requirements. So these were the early domesticates. But when I mentioned about Mendel's, rediscovery of those Mendel's laws in 1900, then selective breeding or conventional plant breeding came into being. Then from these early domesticates, they started making crosses, crossbreeding, as explained here. And then in the process, they were able to develop these modern high-yielding varieties. Here again, the focus was on increasing the production, as Professor Ramachandran mentioned in his opening remarks, to feed the growing population, whatever level that was at that period of time. So idea was to produce more and more grains, to meeting the requirements of the growing population. So these modern varieties, again, you will see here that the red genes, the red squares have disappeared. So that means it says that selective breeding reduces the number of genes or alleles, you call them, or a genetic variation in a population. So these modern varieties, they'll be all of the same kind, and therefore they will not have these properties which were contributed by the genes in red or by these yellow genes. So that's the problem. Okay. So this leaves the population vulnerable to a sudden change in the environment and disease. That's the next problem. That if there is some kind of a disease occurring, devastating, unfortunately, it has not happened. I mean, in the recent past, like we have pandemic, for example, of COVID-19, we were not prepared for it. So I don't want to predict anything at the moment. What I'm saying is there are examples otherwise. There have been famine. There have been famines in the case of potato for a particular disease. There's a late blight disease, and the potatoes were all of this kind. They did not have this kind of variation either in the wild species or in early domesticates, and they all died. So if there is a sudden development of any change or environment, all such plants or crop varieties will be lost. So then comes next to mutagenesis. Now, idea is in these high-yielding varieties, or modern varieties, can we create more genetic variation of this kind? There is a possibility. Scientists have discovered that there is a possibility of increasing this variation here, adding back, for example, yellow or red, or even different colors, which were not here for that matter. You can then do it through mutagenesis, that is developing new genetic variation or diversity by exposing crop plants or high-yielding crop varieties in this case to particular chemical agents or radiation. And until 1920, plant breeding, depending only on the natural genetic variation. This is the natural genetic variation which is available, which was utilized to this process I just explained. But then, this artificial or induced mutation using X-rays in 1930s laid the foundation of creating this kind of a variation through the process we call it mutation breeding, that you can create, because these radiations, and whether it's a chemical mutagenesis or radiation, they have a capability of changing the DNA, at random. We don't know that again. But then while this change must have taken place in the DNA, we radiate the seeds, and then you go to the plant, you see those plants are looking different or better, then you select the ones which are better. Okay, you select still on the basis of the phenotype, we call it, or a particular trait. Then, and one of the earliest examples of this kind is mutation breeding in India, is the dwarf mutant development of rice particular variety by X-ray in 1938, followed by 1960 of a wheat variety, in fact, by Dr. late B.P. Pal and Professor M.S. Swaminathan. They developed this particular variety of wheat, which was on, and it was developed by X-ray mutagenesis at IRA in 1960. So similarly, induced mutations have been used worldwide, since the time it was discovered in 1930, to improve major crop plants, including rice, wheat, barley, cotton, groundnut, beans, and many more. And until today, there is a record in this International Atomic Energy Agency under the FAO that they have got about 3,200 varieties of plants developed using alone, I'm talking about mutagenesis, which is a random process. Still, we do not know what are those changes taking place at the level of DNA, at the level of genes. We do not know that yet. But then, because we wanted to increase their variation and wanted to select the best out of available kind of variation we had, this mutagenesis really delivered, I would say, profusely, and this is a large number of varieties which were developed. But then in 1952s onwards, or in the past, second half of the last century, there were certain scientific breakthroughs took place in science. We call them as advances in science and technology, because science and technology will keep on advancing as we have seen it in the last century and continue doing so in this century as well. And it started with the discovery of DNA as the genetic material in 1952 or 50, discovery of DNA. Now we knew in 1952 that there is a DNA which is genetic material. Genetic material we all knew, yes, there are certain traits governed by the genetic materials, but we do not know what's that compound called, what that chemical is. The chemical is deoxyribonucleic acid. And then what is the structure? Structure was given in 1953 by Francis and Crick, and Watson in 1953 again. So there is a double helix structure of this DNA. Then cracking the genetic code, what does this mean? If there is a structure given, like so there are 4 bases, A, T, G, C, what does that mean? That kind of information was given in 1968. And then recombinant DNA technology took birth for the cloning of genes in 1973. Now we know that there are certain pieces of DNA which act as genes, and those genes govern those traits which we are able to otherwise perceive with our eyes as different kind of traits. So this recombinant DNA technology, for cloning of genes took birth in 1973, followed by DNA sequencing methods. Now they're there, chromosomes, genes, they're all there in a system, in an organism. But if you want to really sequence what comes after A, is it T or G or C and all, because they run into those millions of bases. So that kind of a technology came into being in 1977. We call this a DNA sequencing. And then first method of gene transfer, was discovered in 1983. A gene from a bacteria was transferred to plant by 3 different groups, actually speaking, independently, without knowing that they're trying to achieve the same feat. And they finally all discovered in 1983, the papers were published, that there is a method now developed that the bacterial gene can be transferred. An individual single gene can be isolated. These methods, cloning of genes means only this, that you can isolate a particular gene from an organism, clone it, and try to transfer it to a particular species. For example, in this case, develop the method for transferring the gene from bacteria to plants, in the case of tobacco. This was followed by the scientific advances in genomics in a very big way. We continue happening even today, and in the details. This is now genomics, structural genomics and functional genomics, that you can now, using these methods of DNA sequencing, there are very many other methods now available, next generation sequencing, etc. We call them, that the whole genome sequencing is possible. For example, in 2000, it started with of course, human genome, then went into the crops, like, rice or tomato or potato or any crop you name today, most of them already been whole genome sequenced. So we did not know until, let's say, 1950 that there is a DNA. Of course, and that that's a genetic material. But then you can see in these about 50 years, from these scientific advances have led us to a great discovery, that what the DNA does, it's a gene, and they are gene arranged in a particular fashion. Organizational structure one can determine, the sequence can be determined. So this is the information now which is available with scientists, for example, to develop technologies. I should talk about it, for example, development of transgenic, by transferring these genes. So then genomics, gave us information that now which gene can control which particular trait. Okay, so once you know this, then you will try to play around that gene. Okay, since you have that method available. So, but in some cases still, this slide shows here and trying to emphasize a point that transgenic is not the only answer for each and everything. I'm saying here in some cases, even conventional breeding will be the best method to deploy, various kinds of genes or transfer certain traits from one crop variety to another variety, but then there are certain limitations as well. And in those cases, GM route will be better adopted, where scientists take a gene and insert it directly into a plant, which might be easier, or indeed the only way that which could be deployed for developing a particular trait. And what are those two main reasons that why we need to prefer GM crops over conventional breeding? Number one is that the gene of interest might not exist in a species that can be successfully crossed with a crop. Conventional plant breeding has a limitation that if you want to transfer this virus resistance trait from this particular plant, which is not high yielding, but to a high yielding elite variety, you have to cross. Yes, then you can successfully transfer this gene, and finally you have a plant which is both high yielding as well as resistance to this particular virus. But it takes time. It takes about 8 to 10 to 20 years time, because you have to repeat the cycle, and you have to, when you cross it, all those genes here, they will mix up together with both. So you won't need to restore the genes of all these, actually speaking, except few genes, which we do not know still when we're talking of conventional breeding, what those genes are. But we want to restore most of these genotype, the genes belonging to this particular plant, but only those which are able to give resistance to virus, we want to retain in this. So that takes a long time, actually speaking. So, but then, so this is, but in case you don't have a particular species, that which is resistant to virus, and even if you have a species, for example, there are certain wild species, you are not able to cross it, then conventional breeding will not work. It's as simple. Okay. Or in other cases, why GM route becomes important, that you found a resistant gene in some other plant species. If this is a wheat, you found in tomato, for example, a gene which is able to impart resistance to a virus. But tomato and wheat, you cannot cross. But what you can do using these methods of cloning I mentioned before, you can isolate that gene. For example, if this is the gene, in that particular species, you can isolate this gene now, and this gene could be then directly transferred to this plant. Okay, and this gene that gets inserted, only that single gene gets inserted through the process of transgenic development, and you get this transgenic plant, which is resistant and high yielding at the same time. So therefore, under these two situations, GM route is the only answer. Okay, and while conventional plant breeding will continue to give dividends to produce still the crop plants, with the high yield. Now let's do a kind of a comparative analysis of various methods developed so far. One is conventional breeding, followed by molecular breeding, genetic engineering, or genome editing. Now, conventional breeding is time consuming. I mentioned before, takes 8 to 10 years, even more. And there is a genetic drag of negative alleles from wild species. As I said before, there could be only one gene imparting resistance to a particular virus disease, for example, or any disease. You want to transfer only that single gene, but while making a cross, you have no idea, because you don't know what the gene is. Which one is that gene? We have no idea. They get all mixed up together. So in the process, there are certain genes which are linked to that particular disease-resistant gene as well, they too get transferred, and we cannot separate those two. They are linked. So therefore, this is called, a genetic drag, and that is a problem. And of course, I mentioned to you already about sexual incompatibility, that there are the same crop, for example, wheat, but there is a wild species you cannot, let's say, cross with a modern high-yielding variety, could be of any crop for that matter. So therefore, you can't transfer that gene. Now, second is molecular breeding. This is a process, of course, where we still don't know what the genes are and where they're located, but certain pieces of DNA, we call them as markers, they are linked with those genes of, a kind of a complex trait, So if you can identify those markers, then you can select, based on the bands or the molecular analysis, you can select now, not only based on the phenotype and the field conditions, even the laboratory conditions, you can phenotype them and can try to relate the presence and absence of this marker with a given trait. So it hastens finally the process of conventional breeding. Then genetic engineering. So these two still, we call them as conventional breeding or selective breeding, but this is actually now genetic engineering, which is different altogether. Though conventional breeding will be utilized finally to develop a variety which is commercialized, but to start with the process of genetic engineering, as I explained before, wherein specific genes can be introduced, and the source of gene could be from anywhere, from any organism, because we all know that fortunately, all organisms have got the same DNA structurally, A, G, C, T, those 4 bases, and once you isolate the DNA, just looking at the DNA itself, you can say it is from which particular species. So genetic engineering allows to transfer specific genes for a particular trait into a crop plant. And of course, therefore, it overcomes the barrier of sexual incompatibility. Now, we have entered into a new era, in the last about 1 decade or so, that is genome editing. Okay, here we're talking of precise and targeted genome-wide gene alterations. Here you're taking a full-length gene. There's no other way that you can, try to make any changes. You take a full-length gene, but here you can precisely, targetedly, only few bases, only particular part of the gene. I'll explain it to you as I go further. You can change, and that is, also allows you, therefore, to bring in genetic modifications within the native genes of a given plant species. You don't have to have any gene incorporated from any other species or from any other plant. You can try to still correct the genes which are not functioning properly. I'll give you some those examples. So in short, what I have talked to you so far, what I've tried to mention to you here is that in nature, there's already, actually speaking, genetic modification going on in nature. That is the survival of the fittest. Those plants which are able to survive only will be carried forward by nature to the next generation, and so on and so forth. So this is, we call them as natural transgenics. Okay, then induced mutations, I give example of radiation or chemical changes to increase their variation, that's, but then we have now entered, we're talking about man-made transgenics, where interspecific crosses, if you made it, okay, with human intervention, or you do genetic engineering, or you do genome editing, these are all man-made transgenics. What we're trying to do in the process, we're actually trying to hasten the process. This, first of all, the natural mutations will not be useful for human beings because those are not the traits that you're looking for. Those are the traits which will have, avoiding competition from the next neighbor plant because they need to survive. But what we need to reproduce those plants with higher yield, with better nutrition, better shape, etc., better flavor, So therefore, we need to have human intervention and select only those which we require to have those characteristics in our final end product, make those crosses, or do genetic engineering, or do genome editing. So what I say finally, actually, this is what we're doing is we are only doing plant breeding, nothing else. Whether the nature was doing this plant breeding or genetic modification, or we doing it through the process of whether genetic engineering or molecular breeding or genome editing today, and we are trying to find end products to improve the varieties. At this end, we do not have any knowledge about, first of all, the genes, the DNA. Then we do not have the knowledge which genes were governing those traits. Then we acquired the genes are important for certain traits. We can identify those genes, we can isolate, we can incorporate, and then we can hasten the process of plant breeding. And now we can even do much more. I'll explain it to you using genome editing. So first I'll talk to you about the transgenics, and then I'll talk to you about genome editing. But one more slide, again, to to clarify the fact, to the students particularly, what is the difference between the conventional plant breeding and transgenic route. I have already talked about it, but still to explain to you in simpler ways, consider this is the whole genome of a plant, for example, or plant in this case, and each gene is represented by this particular round circle, of a garland, you can say so. This is a garland, and this is the genome of a plant, for example. This is another plant. This is the commercial high yielding variety. Now you want to transfer this particular gene, which is imparting resistance to this particular variety. You cross the two, both get combined, but finally in the variety you have this gene which you wanted, but you have the genes shown in yellow as well, which we didn't want it, and they could have some bad effects as well. We don't know, but still we try to select the best, a combination of those genes, and finally the new variety will have this gene. You should be happy about it, that it has happened and imparting resistance. But in case of transgenic route, of course, I mentioned before that this desired gene is isolated, gene alone, single gene, and then it is transferred, and finally you have a variety, where this gene gets inserted, but of course it's a random integration in the sense, there are so many, it could get inserted here or it could get inserted here. We have no control over that, but then transgenic route has given a lot of advantages, which you will see as I go further. But then you talk about Indian agriculture, or globally for that matter, what are the challenges? Challenges, of course, we know crop yields are plateauing. The genetic gain or accelerating, is not that kind of accelerating rate we have the moment. Genetic gain is slowed down, okay? And climate change is an issue, which is causing the problem of drought, high temperature, salinity, emerging insect pests and pathogens, malnutrition, as our chairman mentioned, Dr. V.K. Ramachandran as well. Sustainability has become an issue, so that we don't want to damage the environment anymore. We have done that enough. I would say agriculture has damaged the environment enough. And so therefore sustainability is important. And we have finally, of course, the Sustainable Development Goals of United Nations to be achieved by 2030. And according to me, that what is the best solution forward is, I would say, application of emerging science-based tools of genomics and biotechnology to genetically improve the crop plants. You can manage any such trait or any such problem of agriculture with management of fertilizer, timing of fertilizer application, less agrochemical, or whenever it is required. You can manage that very well. But unless you have a genetically improved seed, you can't produce much more to a particular level which is desired. So these are different traits. I don't want to go into the details of all these traits. Actually, idea is that reduce the environmental impact of agriculture so that we move towards sustainable agriculture, and address malnutrition ultimately, and which will enhance, we're talking of, during COVID-19 time, enhance immunity, which we have seen it's so important. So finally, we're trying to manipulate these agronomic traits and nutrition quality with this purpose of reducing environmental impact of agriculture. So these are some of the examples. I'm just giving you one of the examples which I like the most, actually speaking. This is a banana, which is susceptible to a particular disease. We call it as, this is a wilt disease, bacterial wilt disease. But if you take and there's no source of this particular disease resistance in banana. And banana as such, it is a vegetatively propagated plant, okay? So here a scientist, fortunately from India again, of course, in one of the international centers, taken a gene from pepper, okay? And that gene has been transferred using the method of gene transformation, and the banana plant has become resistant, totally 100% resistant. So that's an advantage. So conventional breeding will not work here. So there are selective examples wherein GM route is the best route. And globally, of course, since the time it started, the development of GM crops in 1996, the area has been constantly increasing. And this, of course, I have data only up to 2018 in this particular slide, but it has touched more than 190 million hectares, in area under transgenic, whether you talk of the nations which are developing nations or the ones which are developing. It's almost 103 million hectares or 88 million hectares. So total area is about 190 million hectares under GM crops in the last about, let's say, how many years? About 25 years. And this is the timeline of GM crop production in different countries or commercialization. Started in 1994, actually in USA, and then followed by Argentina, Uruguay, Australia in 1996, again in 1995, of course, Canada. But in India, in 2002, followed by many other countries, until even it continues, the process is on, and there are about 29, 30 countries, growing these GM crops. I mentioned before, 190 million hectares or more, with different crops, about a dozen of those crops, which have been commercialized. In India, I would say, unfortunately, as also been highlighted by V.K. Ramachandran, that so far only BT cotton has been commercialized, and we have harvested enough advantages, I would say. I'll show you some data about that, on BT cotton. And this is the gene which has been taken from bacteria. The BT stands for bacteria, actually speaking. Name of bacteria, bacterial Transgenics, from gene has been taken from this bacteria, inserted in this cotton, and this is a BT cotton. This is different showing here compared to non-BT cotton. Though India has also developed, BT eggplant or BT brinjal, but it has not been commercialized yet. It was available in 2010. In 2017, GM mustard has also become available, but the approval is still pending. But meanwhile, Bangladesh has taken a benefit of the same crop and the event, which has been developed by Indian, private sector, and there they have seen an average of about 20% higher yield or 21% higher revenue, to the farmers, those who have grown BT brinjal. This is a recent analysis. In fact, only yesterday I saw one report, November 9, from one Mr. Vijay Jayaraj, who's from India, has reported it. And that Philippines, for example, is taking a lead much more among developing countries, but to produce more and more GM crops to meet their requirements of food security. But in India, about 5 BT cotton hybrids have been commercialized. Mostly it is a double, gene hybrid of cotton, which is taken the largest chunk. You can see among the approved BT cotton hybrids, which are being grown, by the farmers. And you can see it started adoption of BT cotton in India in 2002 with hardly any area under cultivation, million hectares, but you can see it has constantly increased from nowhere to about 11.5 million hectares, which is almost the total area under cultivation in case of cotton in India, which is, let's say, about 12, or it is 95% adoption by the farmers, of this BT cotton. And here you can say share of this India in world cotton market has also increased. In 2016 data shows that which was about 12% in 2002. Now it is in 2016 it increased up to 25%, which is double. But of course you can see in some cases, for example, in USA it has little come down, or in case of China also it has little come down. But India has really occupied this position of 1/4 share in the international cotton market as a result of increased production of BT cotton. I don't want to go into the further analysis, a lot of information available actually speaking, one can talk about it. But when you talk of research in case of GM crops in India, all sectors, we talk of research institutions, government funded, or universities or state agricultural universities, the National Agricultural Research System, or NGOs, or Indian private sector, or even MNC's private sector. They're all working in different crops, just 10 of them shown here. Of course, mostly you can see the emphasis is on cotton, or on rice. This is, rice and this is cotton, whether you talk of MNC but all of them except few, NGOs are not working all the crops, but most of them, for example, rice, cotton, tomato, brinjal, maize, tobacco, banana, chickpea, pigeon pea, wheat. Pigeon pea or chickpea are so very important crops for India, pulses and oilseeds, and of course, even for food security, we cannot ignore rice and wheat as well. So this is only the data I'm showing you, sharing with you. The GEAC, which is the final apex body for granting approval for the field trials in India. These are the crops which have been granted approval for field trials in the last 5 years until 2020. This is 29 July, about a year ago, 2020, GAC proceedings. And these are the crops where more traits, so far I mentioned about is only BT cotton for insect resistance, but herbicide tolerance or salt tolerance, drought tolerance, frost tolerance, oil quality, abiotic, they're all incorporated in some of these crops, not only by private sector, mostly of course private sector, but again there's the Indian Institute of Pulse Research of ICAR, or you see University of Delhi South Campus, or you see University of Agricultural Sciences, Harvard, and so some of these private, there are more. I'm not giving you a whole list. There are more, many institutions. In fact, when I was the coordinator, we had about 20 more institutions of ICAR, engaged in developing transgenics, and those transgenics are also in the regulatory pipeline, at least 10 of them with different traits, which are not shown here in this particular slide. So far, just the latest data, if I compile all that, I just want to mention that in India, about 16 crops mentioned here, which are under regulatory pipeline with different traits, and events are 83. Means in one crop, for example, in rice, there are 23 different events, could be different traits, or for the same trait, there could be many different events. The events is a single transgenic for the same trait could be different than another transgenic. So therefore, this is about 83 events in 16 crops are under regulatory pipeline. So you can imagine that how much we are waiting, for other than BT cotton, so much information. And crops like even mustard or brinjal, which is a very important vegetable crop for resource poor farmers, chickpea, potato, sorghum, wheat, castor, cauliflower, groundnut, okra, are very important crops in India. Groundnut, okra, again, pigeon pea, rubber, even sugarcane. Though there are only one event in each in these cases, but then they are there. So with regard to, I'm just sharing with you only one slide, and the message here is that more than 20 years that these have been consumed, whether raw or after cooking, different crops. Give an example about a dozen of crops which have been commercialized in different countries in the world, including in India, and BT cotton, which is of course a jute fibrous crop, but I'm sure we all know that BT cotton oil still forms an important part of the vegetable ghee, which is produced in India. And so therefore 20 years of experience and data and analysis shows, that these genetically modified crops are safe for human consumption, for animal consumption as feed, or even to the environment. And I'm referring to, of course, this has been brought out by National Academies of Science, Engineering, Medicine together of USA, including even 123 Nobel laureates that support the fact that these crops are safe. And they say particularly that there's a decreased yield losses, and there's a decreased use of insecticides in BT crops, higher insect biodiversity. Sometimes when you talk of damage to the environment, what kind of damage do we expect? Loss of true biodiversity or land degradation? I mean, agriculture has already contributed enough, I would say, to the degradation of land by irrigating more and more water or indiscriminate use of agrochemicals or fertilizers, the soil microbiota and all you talk about it. So there's, in fact, as such in agriculture, we know it, there's a lot of impact on the environment. But here I'm trying to, really speaking, mention scientifically that these technologies are here to, in fact, reduce that environmental, damage already taking place. So what I was thinking about it, that the environmental damage is increasing as we increase our production. We have done it, let's say in the last century or so. And now we are self-sufficient, even at least India, we're talking about it. We have a right to the Food Act. But now we are in a situation to sit back and think that do we have a technology now to reduce that impact of these technologies on the environment, and can we try to move towards more sustainability, or in a way we call about it organically grown. We have not reached that stage yet. But I'm sure these technologies, if understood well, recognized and adopted well, there's a lot of opportunity, as you will see when I go further. And in fact, this committee report has recommended more public investments to address the issues of food security, through these technologies. This is particularly GEs here talking about genetic engineering. And of course, as a result of, this ability or developing the method of developing transgenics, in different crops, these three were the scientists I was referring to with 3 different papers published in 1983. 30 years later, they got this World Food Prize, and they are Dr. Mark von Mentouw from Belgium, Mary Del Chilton from Washington State University in USA, and Robert Fraley from Monsanto, again, United States. They got this Nobel Prize, sorry, the World Food Prize. Nobel Prize also, of course, I'm talking about recently, given to this particular technology, which is again, advancement, I would say, from genetic engineering, to this genome editing. They got this Nobel Prize, I'm sure you all know, in chemistry in 2020. This is Dr. Charpentier and Dr. Doudna from University of California, USA. And this is a recent slide. In fact, she was making a presentation herself, which I was not able to attend, but I could gather this information just only last month, wherein she said, this is Dr. Jennifer Doudna, the Nobel laureate, who discovered this technology on genome editing. CRISPR, I'm not going through the details of it, but if someone is interested, we can discuss that during question answer session. This is a genome editing technology. Solutions will allow farmers to adapt to a changing climate and sequester more carbon, improving the environment while preserving prized regional varieties or local indigenous varieties. And there is an incredibly important role for these technologies in the production of even small farmers. Until genomic genetic engineering was criticized also that it's only being used more by the larger seed companies or by MNCs because it's a high regulatory cost. As I will go further, you will see that there's not much regulatory cost, as many countries have already adopted them as non-GMOs, any such GM technology. In India, we are still waiting for those guidelines to be approved by the government. Well, I don't know whether I should go into describing this technology, but briefly, I would just like to mention, that now since we know this is the DNA, which is a gene, and if I want to make this gene non-functional, there could be an advantage to the plant. We know that, actually speaking, through the genomics, functional genomics. So if it is made non-functional, then we are deleting the function of this particular gene. And I must tell you, by the way, this is already happening in nature, in one way or the other. When I said there is a natural variation, there is a natural selection taking place, we don't know at what level this is happening, at the level of which DNA or which gene, but through functional genomics and through, now the discovery of various genes and natural genetic variation available in the large amount of germplasm we have in India or globally, we know that there are certain genes which you make them non-functional, that would be good for a particular trait in the plant. I'll show you an example. So here we call it as delete in terms of editing. And similarly, you can add precisely any small piece of DNA and change the existing, or on here, for example, you want to add this to correct the gene function, for example. So you can add this precisely on this location, and that is real speaking edit. Or you insert a new gene, as we normally do in case of transgenic plants. The same thing, you can do it. But here I mentioned before, if you remember, I said that new gene can be integrated at random. We don't know where it gets integrated. But here you can precisely predetermine the location of the genome where you can locate it. So that therefore we call it an insert. So these are, in the parlance of editing, call them delete, edit, or insert. And they can do different functions depending upon which plant with which gene you're trying to do it. Again, this is the same time, Dr. Jennifer Doudna talked about it in October 5, 2021. The same message, I think what our chairman, Dr. Ramachandran, mentioned in his opening remarks, by 2050, the earth will be home to about 10 billion people. And this is something very important, that the food system is responsible for contributing to greenhouse gas emissions to the extent of 35%. So it's already, in fact, deleterious to the environment. This is one example is shown here. So what can we do? Genome editing can help what? It can increase yields and land use efficiency. If you increase yields per unit time and per unit area of land, naturally, that's what Green Revolution did. That we saved a lot of millions and hectares of land, which was otherwise required to produce the same amount of production, and if we had to produce in large piece of land. So therefore, this will increase the land use efficiency, helping farmers adapt to the effects of changing climate, making crop plants more resistant to diseases, and improving nutrition. And particularly to talk about the emission of greenhouse gas emissions, it will reduce those gas emissions from agriculture, for example, methane from rice paddies, or minimizing farmer inputs, which is so important today, fertilizers and pesticides. And these technologies allow you to do that. Okay, and increasing long-term, finally, carbon sequestration in the farm soils, which is very important. By capturing, you can prepare plants which can capture more carbon, which can go through into the system, to the roots, and it could be, helping in gaining the carbon content in the soil. So this is one practical example I'm just showing here. I don't know how I'm doing with the time. Sorry, I'm going a little slow. This is one, for example, the gene. How much time I have, sir?
How much time do you need? 5 minutes? Is that okay?
Yeah, 5 to 7 minutes. Yeah. Minutes.
Okay, because we want to have some discussion as well.
Sure. So this is an example. I'm just showing you a plant which is susceptible, made resistant by deleting the function of a particular gene. And similarly here, this is a plant, rice leaves, which are susceptible to a particular disease, bacterial leaf blight. You can see here 100% resistant, tolerant, these plants have been made. And here, this is an example of rice. This is a non-edited rice. These are genome-edited rice. They are, you can see much better in growth and higher in yield, as you can see here. And similarly, you can do, editing of few genes together, 1, 2, and 3 genes done here. So the ear size, you can see from here to it increases to this size. Precisely we can do that without damaging any other part of the genome. And as you mentioned, Professor Ramachandran, I'm talking about the genome-edited tomato, which has been already commercialized in Japan. This is with the gamma, aminobutyric acid, which is an amino acid believed to be, helping lowering the blood pressure. And similarly, in India, we have this progress in case of rice, making plant tolerant to drought and salinity, or making plant banana enriched with vitamin A, which is beta carotene content, which was appreciated by the then former Minister of Science Technology. This is one of the DBT institutes in Mohali. And these are some of the factors which we're talking about for climate resilience, whether talk of different factors already mentioned about it, which could be improved through genome editing. And one of the best examples here now, which is struck to the scientists, is this. I come back to this particular slide. Now, idea is that, while domestication was important that time, we have been able to develop these modern varieties. But how about if you can now do genome editing in these wild species directly themselves, while you retain these genes, which are good for climate resilience and nutrition, they're there, but then you add into those new genes now, or do editings, or instead of adding new genes, do editing of those existing genes so that you can have more of fruits and better phenotype, etc., and which has been done in case of tomato again. This is a particular wild species which has been converted or domesticated in an accelerated manner, and which has got all those traits of climate resilience and nutrition, and can also have, better yield and better taste and flavor and nutrition content. So actually, this is another point I want to highlight. As you all know, recently this Food System Summit has been concluded, and the message is also there. This global initiatives to reinforce the ambition of science-based solutions will be the key to deliver on the agenda of 2030. Science-based solutions, I think, which is very important. And here I'm just showing you some of those technologies which have been developed, whether you want to develop, tolerant plants to herbicides, or alter nutrients, alter starch content or higher sugar content, or increasing carotenoids, etc. On wild rice, if you can convert in 2 to 4 years into cultivated rice, these are some of the examples, or increase the input use efficiency I mentioned before, whether uptake, transport, responsiveness. So this technology allows you to achieve all that. These are the examples which have already been demonstrated, using the particular technology I mentioned about it. But what is required is, we had a discussion at the National Academy of Agricultural Sciences that a clear policy is needed. And the message is, as a scientist, what I feel, that these crops, whether GM or genome-edited crops, could be an integral component of our self-reliant India. Because we are moving towards more understanding of the nature, more understanding of the complex phenomena of the genes and their gene network together, contributing to a particular trait. And here in many of these countries, actually speaking, these two different categories of plants have been treated as non-GMOs. India, still we are waiting, I mentioned before, but we feel that positive and enabling policies are needed.
What is SDM?
These are actually some of the categories of, I mentioned before, delete, and this is edit. I was in my previous slide, which I can explain you more. So this is in summary that we need more investments for innovations in science and biotechnologies for food security and economic growth. Harness the full potential of genome editing. Science-based regulation is important. It's needed for a meaningful application of these emerging technologies, including genome editing and its variants. More and more actually research being done. Awareness and communication of advances of science to society is required. And that's what I think this particular lecture series is, I'm sure, achieving that particular goal. And then capacity building of researchers and scientists is essential for using this technology in the laboratories for the benefit of developing more crops in India. So this is in brief. Again, I gave you some of those examples, and I'm very happy to deliver this lecture. And I hope I've been able to give you a little bit of a background and idea, that how far we have come, when we talk of improving crop plants, through, kind of a method of trial and error to a method which is well predictable, which is precise and efficient, and that's what. And another message is what is already happening in nature continuously, which is a slow process. scientists, since they have understood the whole process, now can hasten it and can try to achieve what is happening in 20, 30, 40 years of time can be done in less than 5 years of time. So this is in brief. And, if there are certain question answer, I'll be very happy to.
Thank you very much. Thank you very much.
Pleasure.
Thank you. That is an excellent contribution. Thank you to our series. And I'm sure there'll be questions. One question has come. Ramkumar, are you going to curate the questions? Should I start?
I can do that. No, as you wish, no problem.
Have you got it? Yeah.
Yes.
One is, okay, go ahead. If you've started, yeah.
Yes. Professor Bansal, let me start with a question which has come. It says, it has been seen that every year there is a new variety of paddy seed provided to the farmer, and old varieties are almost disappearing day by day. So the question is, why are we losing the old variety of seeds which are rich in nutritional values, less vulnerable to various threats and useful to sustainable agriculture? Is it really necessary to give birth to new high-yielding seed varieties at a very expensive cost? That's the question. That's the first question that has come.
Right. I better take this question and answer right now. Is this what is written by the particular candidate is right? It's just true. And why we are developing these high-yielding varieties? Simply because we want to increase the production. At the same time, we are aware that yes, nutrition is very important. And even those varieties, old varieties, which is or she referring to, they have not been lost. They've been lost, you can say, yes, from the farmers' field. Farmers have stopped growing them because they want remunerative prices and better yield for, increasing production, for feeding the people. It's important. But then this kind of technology, which I'm talking about, it will bring back all those genes which are important for nutrition back to these high-yielding varieties, actually speaking. Or those varieties which he or she referring to are getting lost, which are otherwise nutritious, we can add genes for higher yield in those varieties as well now directly, So ultimately, the idea is to combine the two traits, which is now feasible, and genome editing or even genetic engineering for that matter will be very useful. So the final message here is that no, we have not lost those genotypes. They have been conserved. We have over 105,000 different lines of rice conserved in the National Gene Bank of India. We have, which is the second largest gene bank in the world.
Thank you, Professor Bansal.
Available for us.
Yeah. May I also add, which is of course better known to you than me, that ex situ preservation has been found to be more efficient than in situ preservation of traditional seed varieties. Germination rates have been found to be higher in many ex situ methods than in situ methods. That's Professor Madhura Swaminathan has this question. She asks, can you give a specific example of a gene-edited crop where nutrient use efficiency is achieved so that the application of chemical inputs can be reduced?
Yeah, that's a good question. I'm not aware of any such example currently which has been done in India or elsewhere. But yes, I can figure out those examples. They're there in some of the crops and can send across to you. But certainly, yes, there are cases, wherein with the less nutrient, whether you talk of increasing the uptake more, or they're increasing the utilization efficiency, that, with the availability of the information of genes, which has been done, and I'll be happy to share some of those research papers, but readily I'm not able to give you a specific example where in which crop it has been done.
There's another question, Professor Bansal. It says it's a very broad argument, which more foundational in nature, may I say. There's an argument that GM crops and transgenic crops are against the laws of nature and violate natural biological pathways in plant evolution. What would be your comment?
Actually speaking, how much do we understand the nature? Question is this. And how much do we understand the evolution which is taking place in nature? In nature, we have all evolved from a single species, from a single, organism, actually. And while we have done, genome sequencing of different crops and species, there are genes, for example, in rice coming from other organisms, it's difficult to say that which are the genes which are specific, only present in rice, no other species. So we have all evolved from evolutionary point of view from a single organism, okay? And there have been horizontal transfer of these genes in nature taking place from one organism to another organism, okay? At the evolutionary scale, which we don't understand much at the moment, at least I don't. So the point here is, as long as, as much we try to understand, only then it will be more clear about what is happening. I can only tell you this, that the genome sequencing projects has given us enough information that we have genes from different organisms in a given single species. So we are not disturbing the natural laws of biology at all. If, we have to understand, I don't know how much time we have, but I just give you one wonderful example. And I'm sure this is a very nice, how these plants become susceptible to a particular pathogen. There is a bacteria which sits on a leaf of a rice plant. Now the bacteria will leave a part of its protein it goes into the cell. It will make another protein in the plant which will be eaten, or sucrose, glucose will be eaten by that particular fungus or bacteria for its own survival. So we're all interdependent, actually speaking. We cannot simply say, or so easily, so simplicity. No, I think it's not possible. So,
May I ask a question? May I ask?
Yes, please.
Yes.
I have just a few, actually 3 parts. One is, what are the chances of the government? Are there obstacles in front of the government differentiating between declaring that CRISPR plants, CRISPR varieties are not transgenics? Is that making the distinction like EU? That's number one. Number two, what are the chances of any new plants coming into actual commercial, commercial use? Any new transgenics in the near future? Which are they? Which are they, and what are the real obstacles? Is it scientific, or just the fear of a non-scientific backlash? Number three. Sorry, I have number 3. Is there more hope for CRISPR in the public sector than there was for transgenics?
Say it again, sorry, sir. Third question.
Is there more hope for the development of a robust CRISPR sector in the agricultural public sector than there was for transgenics?
Yeah, I think all the 3 questions are very relevant, and I'm sure it will help people to become more aware about where we stand as India, as a country, on these 3 aspects. Number 1 is, that certainly I mentioned there are different categories of genome editing, and I mentioned 2 of them. I will not be able to explain, but let's say 2 categories, category 1 and 2, they could be easily treated as non-GMOs by any government, scientifically speaking, by anyone here, because what we're doing is we are not inserting any gene even from another plant.
We're editing it,
yeah. So we are only mutating what was happening before. I give you an example of chemical mutagenesis, radiation mutagenesis. They were mutating the genes at random, trial and error. Here we are doing the same thing, however, of a particular gene which is inside the plant itself and doing nothing else. So I think it can be very easily accepted as non-GMOs, and few of the countries have already done it. So I think India should also do it, and we are quite hopeful, as you mentioned. More than
is it close to happening? Is it going to happen soon?
We don't know that yet, but we are working. I must tell you, as National Academy of Agricultural Sciences, we organized about last year a kind of a brainstorming, a stakeholders workshop with both public and private sector and others, even including some farmers. And the message we have given in that particular booklet, which I'll be happy to share with you and Madam Madhura as well, and to Professor Ramkumar, when it very clearly we have proved it in a way, scientifically speaking, based on the evidences we have in the literature. And also when you talk about it, how hopeful we are, what we are asking, actually speaking, I can tell you that very clearly. There's a clause in the Rules 20, in the Rule 20 of Rules 1989 of the Environmental Protection Act 1986. Under that Rule 20, there's a possibility of exemption, that these, I'm talking of these 2 categories, where there's not foreign gene is not there, we're not bringing any element from outside, they should be exempted. And we are quite hopeful. The government is looking into it. Department of Biotechnology, for example, has already accepted it. Now it is under discussion within the Ministry of Environment and Forest at the moment. And we're quite hopeful. And third thing is, you mentioned about what are the new GM crops. There are many of the new GM crops. We have, for example, BT pigeon.
Which do you think, you mentioned which are the new ones, but which do you think are likely to come into any kind of commercial use in the next 5 years? In the next 5 years?
Actually speaking, question is not which one likely to come. Actually, the question is whether any of the GM crops will be accepted. Okay. Once this technology, the gate is open, then I think there are many actually. But question is at the moment whether that gate is going to be opened by government or not to accept more of GM crops other than BT cotton. We have, of course, GM mustard hybrid. We have got BT brinjal.
That's what I asked you, sir. So is the obstacle scientific? Is it from the GAC, or is it the fear of the opposition? A fear of is it concern, not fear, concern about uninformed opposition, or what is it?
No, I think this is more to do with some kind of a social acceptance or public acceptance of some of these GM crops, and probably there's not enough communication of science, to the public at large, or otherwise, if you're talking from one scientist to another, I'm sure we all convinced very clearly, since we understand the technology very well. And I repeat that what has been happening in nature, same thing, the scientists now have discovered the ways to repeat it in a different and efficient manner. And there's no other, and there could be, in fact, there are methods, if you talk about any kind of damage to the environment or other, there are methods of conventional bred crops, for example, potato with more alkaloids, there are many other examples of this kind which have got anti-nutritional factors in them, but then those now anti-nutritional factors could be even corrected using these technologies. So what I'm saying is that we have to have the pros and cons, and not you go blindly, you go by case basis, and there are, I use the word relevant GM and genome edited crops. That by that I mean is that anything which you cannot achieve by conventional breeding, and you need that trait, for example, BT cotton, I know BT pigeon pea or BT chickpea in our country, we're losing, so much out of the damage because of these insect pests in case of our pulse crops, particularly, or in rice for that matter. I think so. If you cannot achieve that goal, which we have not been able to do so far, otherwise we could have done it by conventional breeding. We have done a lot, So at least in those cases, we should adopt GM crops or even genome editing, even much more safer than even genetic engineering technologies.
Thank you, Professor Bansal. There are 3 more questions which are there. One is from Charu. She asks, gene flow from crops to wild or weedy relatives is often discussed as a potential risk in the commercialization of transgenic crops. Is there actually a real risk of a transgenic gene migrating or passing from a GM crop to a genetically similar mimic weed or its wild relative? That's the question.
See, this is scientifically, this is possible that genes do get transferred from one species to another. And that's what I said before is happening in nature, But then the question is, you're talking here now of transferring of genes from a particular crop back to a wild species or a weedy or wild relative, Now the question is here, first of all, we know on the global basis that for which crop, where are the wild species grown? For example, in India, we have compiled the whole information, And so we have to find out if we are trying to release a particular crop in India, let's say, for example, brinjal. Do we have those wild species or weedy species of brinjal in our area where the BT brinjal is likely to be released? Yes, they're there. They're there. But experiments have been done that those, even by making crosses by human intervention, those are not crossable, those species. I said before, gave you an example of sexual incompatibility, even the same crop species. And even if they are able to make crosses and you get a seed, hybrid seed, it will not form, the seed, fertile seed will not be formed, it will not be able to germinate. So it's not easy again, at times even scientists try to take a gene or make a cross between wild species and a crop plant, they're not able to successfully do it, So there are other reasons, scientific reasons that you cannot do it. So there are many other factors, we cannot simply say that yes, or we cannot say this, no, but so far, whatever has been commercialized, there's no such example which has come into focus.
Thank you. There is yet another question from Vivek Babu, where he basically asks, he fears that advent of the BT cotton crop in India has not quite reduced pesticide use dramatically, because with BT cotton, with 95% usage among farmers, why is cotton crop still one of the top 3 pesticide consuming crops in India? How collective institutions can tackle the resistance to BT toxin. Like, for example, cultivating refuge crops should be mandatory, but it is not being done on the ground. So what would be your response?
These are valid points. Okay, I'll take one by one. First is, it's not that the pesticide use has not decreased. It has decreased. There are enough data. I'm talking about 2002 onwards. I'm not just comparing only looking at this year now, 2021 or 2022. We've got 20 years is a long time, so quite possible. And it is true, that the use of pesticide has come down, number one. Number two, the resistance has also been reported to be developed, by these insects against a particular tool, And number three, when the plants became resistant to a particular pink bollworm, it has become susceptible to other pests. This is a continuous phenomena. It's a very dynamic behavior of the organisms, which are thriving on a particular plant species. So they keep on mutating themselves. They keep on becoming tolerant to resistant. But however, if you look into the long history of 20 years, is not a short period of time, for example, or the gains which we have seen, immediately, and there are enough data available to show. So one has to see the balance where we are. We are towards the plus than in the minus. And this we have to understand scientifically, that these organisms will ultimately develop resistance, yes. But then we have to therefore find more genes. And to talk of durable resistance, therefore, we have got 2 genes, for example, BT and cotton hybrid, which is, I think, Bolgar 2. So that is only to delay the resistance development, not to delay the resistance development by the insects. So this is a continuous, because we're talking of living organisms here, they're trying to find their own ways to survive. So I think, but we have certainly harnessed a wonderful advantage, I would say, from 14-15 million bales in 2000 or 2002, I believe, we've gone up to 35 million bales, So certainly there, and then the quality of the produce, one has to see. I can understand there must be other pests now coming in, we're talking of mealybug and few other things, other insects coming in. But we have already harvested the advantage, I would say, quite a bit, So we have to appreciate that part as well. And that allows to even develop more resistant and robust, resistant plants.
The next question is again by Vivek Babu. He basically asks, what are the applications, if any, of the CRISPR technology in green manure crops and cover crops? Because they're also a very important plant species, and whether CRISPR can help in that kind of crop growth or crop development as well.
Yes, there is a very big possibility there as well of using this CRISPR. But we have to see that how much genomics work has been carried out, actually speaking, in those crops. This is certainly, you can say, a bottleneck, of using these CRISPR or genome editing technology, that you have to generate, and technology allows you to do that. But the question is whether we have done it or not. If we have not, we better do it. Whether we have enough information on the genomics of that particular species or not. If we have that enough information available, I'm sure there are plenty of opportunities for increasing the biomass or accelerating the development of the plant. There are very many possible ways by which you can improve even those forest crops and other crops.
Yeah.
We have that information. Technology is there, yes.
Just to add, China has used this BT technology substantially in forestry. BT poplar, for example, is a very important innovation in China, which is used in more rapid afforestation of the Chinese countryside. There is one question by Prachi, where can you comment on the legal framework governing GMOs like patents, IPRs, etc.? Is the Indian legal framework responsible for less dissemination of these crops commercially?
No, legal framework we have already, I said before, the Environmental Protection Act governs this, which is 1986. And there are very clear guidelines there about that Environmental Protection Act. As far as GM crops are concerned, there's no doubt. We have got, actually speaking, I would say, a wonderful regulatory system under this act. And these rules, 1989 are being followed thoroughly. And as a result of those protocols issued by a safety committee, review committee on genetic manipulation, and then GAC, and they're all in place. But yes, there's a question of, intellectual property attached to it, So that is something we have to all worry about it. And I think when we want to, popularize these technologies or want to make use these technologies in India more and more, we have to take care of two things. One is that we must try to invest as much as possible in these technologies for the development of them or using them on a large scale for more number of crops. And second is somehow we have to have freedom to operate, with regard to the IPR attached to these technologies. And investment is leading, I mentioned before in my summary slide, for basic sciences, so that we have our own patenting right on some of the novelties and the methods, which have been, which can be developed in India. I give an example of Debojyoti Chakrabarti, for example, one of the young scientists who is doing wonderfully well. He has, found his own Cas9 one gene, which is required, to do the genome editing. While we can have the information available, which is already done by others, but then you can do more on your own and generate your own IP. And actually speaking, therefore, more research and more innovations and IPs, investment is required for this.
Thank you. There are a few questions which probably you have already answered, which are related to the loss of biodiversity and so on with the coming of BT crops. I'm going to skip them. There's a question by Gautam here, where he says, there has been an effort from several international agencies, including the FAO, to push for the so-called nature-based solutions, including, for example, agroecology and so on. That's my addition to that statement, especially in the context of climate resilience, which rely purely on agroecological methods with minimal technological interventions. How can one reconcile both these approaches being pushed by international agencies?
See, this is right that we can try to manage, the climate problems or other, environmental related changes with some of these indigenous technologies, no doubt. But we have to always remember that, we have to produce more and more from less and less land. We cannot go for deforestation. So if you want to do whatever you want to do, for example, with regard to climate change or nutrition in a much more efficient manner, and for larger population, we're talking about always purchasing power increasing and kind of a diversity we want to further increase of the available food crops. So I think one technology alone may not be enough. And yes, there is a possibility of reconciling the kind of genome editing technology we're talking about it, because finally the produce which we're talking about is a crop which is being, propagated through seeds, so the genetic component of the seed and the environment both have to interact together, to have a final product. So one, environmental technologies or kind of processing or cultural practices alone may not do the job, or seed alone may not do it if it is not given that particular environment or is not prepared with inbuilt resistance to thrive in that environment. So I think, yes, both can be reconciled easily, and therefore, I think the efforts are in progress, I would say.
May I, Dr. Bansal, I think there's, before we have to wrap up, I think there's a concern that Gautam has expressed that, which not being addressed, isn't it disappointing to see, for instance, things like ZBNA and all that coming in FAO documents? I mean, it's very so, and I think that's what he's talking about, that some of these, one step away from open charlatanry coming, one would have expected that in this period of debate, FAO would have come strongly on one side. Isn't it a matter of great concern that these scientific earlier it would have been FAO which was spearheading this, the scientific alternative. Nowadays, unfortunately we're seeing we wouldn't be surprised if these so-called what Gautam called nature-based solutions, are being, as if these are two things which deserve equal time.
Yeah, you're right, but I would certainly advocate still the use of this technology. I'm talking about it as part of the nature-based solution.
Yeah, exactly. Yeah.
Because please remember that we are trying to harvest more with less energy, with less time, with less land.
Correct.
resources. And I tell you, we have got, and we are not discovering anything new. Already, as someone mentioned, that we have those nutritious plants which are not being accepted by the farmers grown. They are conserved. Any kind of natural genetic variation which is available, we are trying to only mimic it, through these genomic technology, by understanding what are those changes that plant making so, tolerant to, for example, saline water. What are those changes? Can you bring those changes to a cultivated plant?
Correct.
Right. So these are all nature-based solutions only we are moving towards. And certainly I would say that these technologies are certainly part of these nature-based solutions.
Yeah, this is our agroecology, let's call it. But
Yes.
I think I think we have answered almost all the questions, and I must pass on back the mic back to Professor Ramachandran. Yeah.
No, but before just another minute, I will try to steal here, because somebody mentioned about the biodiversity, BT cotton you mentioned. I must make it clear that the population of honey bee in India as a result of BT cotton cultivation has increased. And I don't have to give any scientific reasoning to it. I'm sure any common person will understand it, that by indiscriminate use of pesticides, any insect will get killed. Specific technology only to kill that particular bollworm, which is damaging the cotton bomb. Therefore, even other plants, other bugs are coming in. That remember that they are not getting killed. So how can honeybee will and that is fair. Therefore, that population is increased. There are scientific experiments done by certain universities.
Thank you very much. That is thank you for really excellent contribution, and I come to our series. And as was mentioned earlier, this is only the first of your the first of your interactions in a seminar, or in this case a webinar with the FAS. We look forward to much more, and to your sharing your expertise, knowledge, and experience with us, and helping us in as social scientists in our quest to find scientific solutions for increasing agricultural products supplies, in bettering livelihoods, and generally in improving people's opportunities and incomes throughout the world. Thank you very much. Thank you. Can I hand it over to the director of the FAS? Is any?
Thank you, Professor Ramachandran, and thank you, Dr. K C Bansal. And it was we are very grateful that for this lecture, and as Professor Ramachandran just said, that this is just an indication. This lecture series, in fact, is an indication of our intent to continue our quest to understand the importance of applying modern science and technology for the development of agriculture, in terms of productivity, in terms of profitability of those who are dependent on agricultural production, and in terms of sustainability of the environment and ecology. And I think the note at which you ended, that these solutions which are science-based are also nature-based solutions. So expanding our understanding of nature-based solutions is something that perhaps we in the social science community would sort of benefit a lot from. So I can only hope that we will continue this conversation on various occasions, and I would certainly hope to meet you in person in the next workshop or conference that we plan to hold shortly. May I now hand over the mic to Mihira for the official vote of thanks. Mihira.
Thank you, Sandeep. I would like to thank all the participants who have attended today's webinar and who have contributed with their questions. Please note that our next lecture will take place on November 17th. The speaker is Dr. Himanshu Pathak, and he will be speaking on abiotic stress management. I'd like to thank Professor V.K. Ramachandran, our Managing Trustee of the Foundation for Agrarian Studies, for agreeing to chair this session today. I would also like to thank Professor Ramkumar for managing the question and answer segment. You can find the inaugural lecture that Professor Ramkumar delivered for this series on our website, fas.org.in. I would also like to thank our director, Dr. Sandeepan Bakshi, and also Rosa Luxembourg Stiftung South Asia for collaborating with us on this series. And lastly, I would like to thank Dr. K C Bansal, sir. Thank you for agreeing to speak and really enriching our series with your knowledge on transgenic and CRISPR technology. And this recording will be saved for future uploads to our website. But if anyone has any other questions for Dr. Bansal, you can please write to us. And we will forward them over to him and share any material that he would like to share with the attendees and the scholars who have attended today. So thank you, everyone. I will now end this lecture. Thank you.
Thank you. Thank you very much. Thank you, all of you.
Thank you very much.
Thank you. Thank you, Madam Madhura, Dr. Sandeepan, Professor
Thank you. Thank you. Excellent. Very, very excellent.
Thank you very much. But I
Thank you.
me too.
That is,
Yeah. Great.